started. We have Katrin Stuber from the University of Copenhagen. Katrin received her PhD under the mentorship of Martin Scheffner and Andreas Marx at the University of Constance, where they established a system for incorporation of non-canonical amino acids to site-specifically phosphorylate proteins, and thereby study phosphorylation in the context of ubiquitin proteasome system. Also really interested in how phosphorylation and ubiquitination works in mitophagy. They use biochemical and structural biology to answer their questions. Here they became intimately familiar with affinity enrichment to study phosphorylation-dependent interactions of proteins, and also the use of mass spec. They are now doing a postdoc with Jesper Olsen at the University of Copenhagen, where they are investigating high-throughput phosphoproteomic drug screening in the context of triple-negative breast cancer to identify novel targets for rational drug design and the generation of combinatorial therapies. And looking through their CV, they seem to have won every prize that they have competed in, from best poster to best talk to best paper. And today they'll be talking about studying structural and functional consequences of site-specific phosphorylation and ubiquitin and NEDD8 by using non-canonical amino acids. Take it away, Katrin.
Thanks for the introduction, and I just have to find my pointer here. Yeah, and of course, thanks for the opportunity to present my work in this webinar series today. And yeah, as I said, I would like to talk about studying structural and functional consequences of phosphorylation in ubiquitin and NEDD8, of course, by using unnatural amino acids. But before starting with the unnatural amino acid part, I would like to give you a short introduction into ubiquitin and ubiquitination. You might know that ubiquitin is a small protein that is attached post-translationally to substrate proteins via a 3-enzyme cascade. So we have here the E1 enzyme that activates ubiquitin. It is transferred to the E2 enzyme. And in a final step, the E3 ubiquitin ligases transfer ubiquitin to the lysine residue of a substrate protein. And ubiquitin can also serve as its own substrate, leading to the formation of ubiquitin chains. And these chains can have different topologies, and depending on the topology, this has a different cellular outcome. And the most prominent one is, for example, the lysine 48 linked ubiquitin chains target proteins for proteasomal degradation. But yeah, it was approximately 9 years ago, it was found that the modifier ubiquitin itself is also subject to post-translational modification. It can be phosphorylated, but also acetylated, which will Simon cover in his talk later on. But I'm focused on the phosphorylation now. And ubiquitin, you can see the structure here, has 3 serine residues, serine 20, 57, and serine 65, and all of them are known to be phosphorylated, so they have been found in proteomic studies to be phosphorylated. And how is a phosphorylation, of course, installed usually in a cell? You need the kinase that, under the consumption of ATP, transfers the phosphate group onto a serine, threonine, or tyrosine residue. And of course, this post-translational modification can also be reversed by the action of phosphatases. But to study a site-specific phosphorylation in a protein of interest, in this case ubiquitin, you need to have the kinase, and this has to be specific for your site. And for example, for position 20 and 57, the kinase is not known. So we made use of the amber stop codon suppression technique, so to incorporate unnatural amino acids to site-specifically phosphorylate ubiquitin at these 3 sites. And the system that we used was the system published by Jason Jin in 2015, and we cloned the tRNA and the tRNA synthetase onto a plasmid, and we supplemented the expression media with phosphoserine, and this enabled us, together with the incorporation of the amber stop codon at the desired position, to incorporate phosphoserine site-specifically at these 3 sites. And I guess when you follow this webinar series, you might know that phosphoserine is not an unnatural amino acid per se, because it is actually the precursor to serine in the biosynthesis pathway of serine in bacteria. And this is why this incorporation system usually comes with a special expression strain in which this phosphatase here is knocked out to have high levels of endogenous phosphoserine to have a better incorporation. Furthermore, we of course were interested in incorporating the non-hydrolyzable version of phosphoserine, which you can see here, where the phosphate group is exchanged by a phosphonate group, and this is resistant to phosphatase cleavage, which is really important when working in cell extracts. And for the incorporation of this amino acid, we would like to actually have the complete opposite. So we would like to have low levels of endogenous phosphoserine, which is achieved by the overexpression of this phosphatase and using a expression strain where this aminotransferase is knocked out. So first of all, we did some test expressions for these 3 sites, and we found out that it is highly dependent on the site how efficient the incorporation of phosphoserine is. So on position 65, we can see we get nice full-length product here detected by a C-terminal his tag. However, for position 57 and position 20, we can barely see a full-length product in the anti-his Western blot. And here we were asking, is it the positions per se that are bad for amber stop codon suppression, or is it the negatively charged phosphoserine that makes a problem? And this is why we incorporated propagulycin at the same positions, which is a system that we also commonly used in the lab, and we know that it works properly. And what we could see was actually that this PLK is really nicely incorporated at position 20 and position 57. And we get even a little bit less of incorporation on position 65. So we see the reverted picture, meaning that it is not the amber stop codon suppression per se that is a problem on these sites, but it seems to be this incorporation of this negatively charged phosphoserine during translation in these 2 positions, which makes the problem. But of course, it is always a problem to get high yields with these systems, and especially with the non-hydrolyzable phosphoserine. And because, just to give you an impression, we had to synthesize it on our own with a simple 3-step synthesis. But of course, it is very precious material, but you need quite a lot. So for 1 liter of expression culture, we used 1.46 gram of this non-hydrolyzable version. And this is why we wanted to improve, of course, the yield. And this is what I got out with this delta C knockout strain, which was 0.5 milligram per liter culture, which was really not great. And this was why I just tested then the different available expression strains in the lab, which was BL21 and B834. And surprisingly, with this B834 methionine oxytrophic strain, I got a 4-fold increase in non-hydrolyzably phosphoserine incorporated ubiquitin. And this is why I then went on with this B834 strain for further expressions. Now you might ask, here we don't have this knockout of this C, so we have endogenous phosphoserine present. Yes, but we could overcome this in ubiquitin with a purification strategy that looked like that. So we have our C-terminal his tag to get only the full-length product. We can tracelessly remove the his tag by this deubiquitinase, And as a last step, we then have an anion exchange chromatography with a pH gradient. And this is how it looks. So you can nicely see, so this is a phos-tech gel, and phosphorylated proteins migrate slower than the unphosphorylated counterparts. And you can also see that the non-hydrolyzable version runs at a slightly higher height than the natural phosphorylation. And you can see here that we can, with this pH gradient, get a baseline separation of non-hydrolyzably phosphorylated ubiquitin and the naturally phosphorylated ubiquitin. So we could get really pure protein out of it. And in the end, we could create all of these ubiquitin variants phosphorylated at position 20, 57, and 65 with the natural phosphorylation and the non-hydrolyzable phosphorylation. And if we add phosphatase here, we can see that it's nicely stable. Whereas for the non-hydrolyzable variant, we get the shift to lower molecular weights. However, the yields for these other two sites were really low. And so we did some investigations on E1, E2, E3 with these ubiquitin variants, but I won't show this today. I will go on with the question, is the non-hydrolyzable phosphoserine analog a good surrogate for the real phosphorylation? Because what is usually done is to use aspartate and glutamic acid as phosphomimetic mutants. But yeah, already from looking at the structure, they have some disadvantages, I would say. And what we looked at, I give you some background to this. So this phosphorylation in ubiquitin at position 65 is known to induce a second conformation. So you have usually this normal conformation of ubiquitin with serine 65 phosphorylated, but upon phosphorylation here, you induce a second conformation in which the C-terminal tail is retracted into the core of ubiquitin, and this serine 65 containing loop is extended. This also disrupts this important interaction site of ubiquitin, and it's really a huge structural rearrangement. And since this is a really important feature of serine 65 phosphorylation in ubiquitin, we were interested if this holds true also for the non-hydrolyzable variant. And interestingly, I have to say, this second conformation was not seen in NMR studies with aspartate or glutamic acid mutants of the serine 65. But strikingly, we could see that with this non-hydrolyzable phosphoserine variant incorporated at position 65, we get this retracted conformation. And if we compare in NMR structures of the natural phosphorylation to the non-hydrolyzable phosphorylation in the relaxed state, we only see some variants here around serine 65, whereas if we compare the retracted state of the natural phosphorylation and the non-hydrolyzable phosphorylation, they are really super similar. So they resemble each other very well. In terms of population, so for the natural phosphorylation, this retracted state exists in 44% at pH 7.4, whereas for the non-hydrolyzable analog, we only have 12%. And we explained this fact by the fact that the non-hydrolyzable analog has a higher pKa, and it was shown that this double deprotonation of the phosphoserine is important for this retracted state. And so we concluded that this might be due to the higher pKa of the non-hydrolyzable phosphoserine. But on a structural level, it resembles the natural phosphorylation quite well. Then we also looked at it on a biochemical level, of course, and here we used an assay that was published by the lab a few years ago, and it monitors the E1 activation of ubiquitin, so the first step of the ubiquitin cascade. And what we can see that wild-type ubiquitin is nicely activated, Whereas if we look here at red, the phosphorylated ubiquitin is less well activated than the wild type. The same holds true for the non-hydrolyzably phosphorylated ubiquitin. Whereas if we look at the mutant D, that behaves like wild type, and the mutant E is somewhere in between. So also here on a biochemical level, the non-hydrolyzable phosphoserine resembles the real phosphorylation better than these D and E mutants. And we could also show in E3 ubiquitination assays that phosphorylated ubiquitin and non-hydrolyzably phosphorylated ubiquitin behave the same and are less well used for ubiquitination of E6AP in this case. So overall, we could show that on a structural and biochemical level, the non-hydrolyzable analog is really good resembling the real phosphorylation. And now I would like to talk to you about NEDD8. NEDD8 is also a ubiquitin-like modifier, and it is the closest relative to ubiquitin. They share 60% of their sequence, and they are 80% identical or similar. And most importantly, both of them have this serine at position 65. And interestingly, if you look at phosphosite.org, also this serine 65 at from NEDD8 was found to be phosphorylated. And this is how we got into the investigation of NEDD8 65 S65 phosphorylation and what it is. And what we could show is that NEDD8 phosphorylated at position 65 is also phosphorylated by PINK1, which is the kinase known for this phosphorylation site in ubiquitin. We could show that phosphorylated or serine 65 phosphorylated NEDD8 increases the phosphorylation of Parkin, and we could show that phosphorylation of NEDD8 or phosphorylated NEDD8 can also activate the E3 ligase Parkin, like it was only shown before for ubiquitin, for phosphorylated ubiquitin. And the next thing we were asking, of course, was, is there also this second structural conformation present in the phosphorylated NEDD8? And to do so, we again produced our phosphorylated NEDD8 with stop codon suppression, but this time also heavy amino acid labeled. So this is the NMR spectra of wild type NEDD8. And if we overlay now the spectra of phosphorylated NEDD8, we can already see that we get approximately twice as much signals as before for the wild type, which already indicated there might be a second conformation. And then by creating a 15N 13C labeled phosphorylated NEDD8, we could really solve this, or especially Tobias Schneider, which was my collaborator at the university back then, could solve this, that we really have also these two conformations. We have this relaxed state that only has perturbations in the vinicity of serine 65, and we have this retracted state, which has perturbations of the structure all over the sequence. And when looking at the secondary structure analysis, we could also see that this retracted state is induced by a retraction of the C-terminal tail into the core. So we have also this slippage of the beta-5 strand, as it was only shown for ubiquitin or for phosphorylated ubiquitin before. And of course, also the retracted and relaxed state here are populated almost equally at pH 7.4, as it was shown for ubiquitin. Now, what do we use these? So far, I mostly talked about the normal phosphorylation, and but I showed you that these non-hydrolyzable analogs are really a good mimetica. And now I would like to show you what we use them for. And we actually use these non-hydrolyzably phosphorylated variants to identify the phosphorylation specific interactome of these proteins. And to do this, we generated these variants with an N-terminal strep tag. So we have the ubiquitin with the phosphorylated ubiquitin, the non-hydrolyzably phosphorylated ubiquitin, and the same for NEDD8 with an N-terminal strep tag as a handle. And with these baits in hand, of course, we also included the non-phosphorylated counterparts. We then incubated them with cell extracts, and then we washed out unbound proteins. We eluted the bound proteins and triptychally digested them and analyzed them by LC-MS and looked then at the data. And we did this with two different cell extracts, but we could find almost the same picture. So we get two distinct interactomes for ubiquitin and NEDD8, as you would expect. And more interestingly, you get also these clusters of specificity within NEDD8 and ubiquitin. So what you can see, for example, here is proteins that are specifically enriched in non-hydrolyzably phosphorylated NEDD8 compared to unmodified NEDD8. And these were mainly HSP70 proteins. Then here we have a cluster with NEDD8 and phosphorylated NEDD8 preferentially enriched. And this is a cluster which contains a lot of proteins that count to the denedylases. And in this cluster here, in which the proteins have preferentially bound to unmodified ubiquitin, we find almost the whole proteasome, indicating that phosphorylated ubiquitin is not used as a signal for proteasomal degradation. And down here in this cluster, we have proteins that don't have a preference for modified or unmodified. And here we find, for example, a lot of ubiquitin ligases. But of course, we were most interested in these interactors of phosphorylated ubiquitin and phosphorylated NEDD8. And we had a closer look at these proteins. And what really stood out here was this cluster of HSP70 proteins that were highly enriched with the non-hydrolyzably phosphorylated NEDD8. And when looking into literature, we found that it was described that NEDD8 can activate the ATPase activity of HSP70 proteins. And we then performed also this kind of assay, and we could show that indeed NEDD8 can activate the ATPase activity of HSP70, but phosphorylated NEDD8 can stimulate HSP70 ATPase activity even more. And this also holds true for the non-hydrolyzable analog, of course. And overall, we found a lot of proteins that are involved in different kind of stress responses here. And this led us to the conclusion that phosphorylated ubiquitin and phosphorylated NEDD8 do not need to be covalently attached to exert their function, and that the phosphorylation of ubiquitin and NEDD8 plays a more prominent role in different kind of stress signaling pathways as previously assumed. And closing the loop now, there was a really interesting study published where they looked at the interplay of NEDD8 and Parkin in the context of neuroinflammation. And they saw that under neuroinflammatory conditions where interleukin-1 beta is secreted, NEDD8 is translocated from the nucleus to the cytoplasm. And they also found that Parkin and NEDD8, that they co-localize. And then they did MD simulations and saw that NEDD8 could potentially bind to this known phosphoubiquitin Parkin binding site. And this is why they concluded that under neuroinflammatory stress conditions, NEDD8 activates Parkin. But with our studies and what we did, we could show that not NEDD8, but phosphorylated NEDD8 can activate Parkin. And this is why we hypothesized then that under neuroinflammatory stress, not NEDD8, but phosphorylated NEDD8 activates Parkin. And actually this finding was the basis for the postdoc proposal I then wrote, and also recently then got the Marie Curie postdoctoral fellowship for. And yeah, this is what I will investigate further in Jesper Olsen's lab, where I will look at the proteome and phosphoproteome changes under neuroinflammatory stress conditions. And yeah, with this, I would like to thank the people in Constance, because what I showed you was all things that I done in Constance back then, and especially Tobias Schneider, who did all of these NMR measurements and interpretations. And yeah, then of course the new lab, and you for your attention.
That was great. Thank you. People can ask questions through the Q&A, or I can give access to asking through Zoom. I would like to start off a question, and maybe I missed this, but can you take your protein that you phosphorylated and like electroporate it or deliver it to cells, and see if you can get interactions inside the cell or drive signaling pathways rather than looking at lysates?
Yeah, actually we tried something like that with a it's like a microfluidic chip where you then squeeze the proteins in. So we used it before mainly for poly-ADP ribosylation, so NAD analogs, to get them into the cells. But we also tested it with proteins at one stage, but that never really worked super well, because this is a lot of stress you induce to the cells. So I think in that case, it is maybe better to work with the lysates, or then use the incorporation system in mammalian cells.
Yeah, so it's a delivery problem, right?
Yeah.
Can I ask a quick question? That was really great. Thank you, Katrina. That was really fun to hear that story again. I really appreciate the fact that you did those comparisons of phosphoserine proteins with the non-hydrolyzable. We've done a lot of that too, and because we've got a lot of pushback from the community because of that. But I had one question for you. So you said you attributed the difference to the pKa of the two, the phosphate versus phosphonate, and
Yeah.
I was curious, did you try, say, doing that same analysis at slightly higher pH, say like pH 8, to, you know, try to deconvolute that a little more? And if not, do you think that any contribution came from the lack of the hydrogen acceptor atom, the oxygen versus the methylene group? Could that have contributed as well?
So to the first question, we never did it at a different pH. We only did it with the pH 7.4. And of course, it could also be this the second, yeah, it could also be something different than pKa. But this was what we thought would be the most most conclusive explanation for it. But of course, we never digged really into it. What is the cause for that? Of course, some people also use this fluorinated version of it, which should be the same pKa, but of course then it's much more difficult to synthesize it, and you cannot use your permafrost system anymore. So yeah.
Oh, thank you.
There's one quick question. Have you tried microinjection? Electroplating proteins is super limiting, so just microinjecting your protein into cells.
No, we never tried that.
Philip. Okay. But good. That is a surefire way to get proteins in.